Hello, my name is Michelle Atallah, and I'm a PhD student in the lab of Dr. Parag Malik at Stanford University. Today, I'm presenting ImmunoGlobe, enabling systems immunology with a manually curated intercellular immune interaction network. Pathway and network analyses have long been very valuable tools for understanding complex multi-step mechanisms in many different fields of biology. Whether it's cancer signaling, gene regulation, or metabolism, these network analysis tools have helped us make sense of the large data sets that we collect. However, no such tool yet exists for the immune system. Now, the immune system is a complex, highly interconnected system made up of several different immune components. And while recent technologies such as cytoph and flow cytometry have allowed us to profile immune responses in significant depth, we're not yet able to extract mechanistic understanding from this data. And without mechanistic understanding, this information is not very actionable. We can characterize diseases and sometimes even classify patients based on whether their immune response is effective or not, but we don't have the mechanistic insight needed to precisely modulate immune responses in both health and disease. So the first part of my project was actually building a network map of the immune system that we could use as the basis for these types of network analysis tools. We started with Janeway's Immunobiology, which is a classic and pretty comprehensive immunology textbook. And we went through and codified all of the interactions described in its text, figures, and tables. For each interaction, the minimum information required was the name and type of the source and target nodes, the type of interaction, and the text reference that it came from, with additional information collected as it was available. This is an example of one sentence from the textbook and how it was codified. This single sentence contains 7 individual interactions, each of which gets its own row in the network table. And you can see here that the arrows indicate the interactions. The arrows originate at the source node and end at the target node. They're numbered according to the table in part C. And then the network table was used to generate a graphical representation of all of the interactions that we codified. So the sentence in B and the network graph in part D both contain the same information, but part D is much easier for us to understand the relationship between the components. That's true both visually and because it's a graph, it becomes much more computable. There are types of analyses that you can do on graph structures that you just can't do on unstructured text like that sentence. So we went through the entire textbook and codified every interaction that it described. And this is the result. So we have this network of 253 nodes, which represent all major components of the immune system. The node types are shown down here, and the edge types are shown in this legend on the right. And this is how it's organized. So the different components are grouped according to their type. So looking at the network structure, we showed that it recapitulates known features of the immune system, kind of just as a sense check to make sure that it reflects what we know about how the immune system works. So 253 nodes, 1,112 unique edges among them. Despite this large size, the density of the network is only 0.02, which is pretty low. That makes sense because the immune components do need to be specific. When you mount an immune response, that response is very specific to the type of pathogen that you've encountered. So whether it's bacteria, viruses, parasites, that all initiates a different type of immune response. So the low density reflects that. However, despite the low density, the average path length is pretty short. And so here you can see the different types of interactions that there are. Most of them are between immune cells and cytokines because they are the major players of the immune system. And this is what, again, an example of what the network looks like. So it looks pretty dense, but it's not based on numbers. The average path length of 3.25 means that if you choose any 2 nodes at random, it takes only just over 3 steps to connect them. This also makes sense because despite the low density reflecting the specificity of the immune components, the short path length means that the immune system can very quickly disseminate information across the entire network. This is especially important in the context of pathogenic infection, as we discussed, because timely responses are crucial to containing the spread of infection and mounting an effective immune defense. So we verified also by generating random graphs that this short path length is not something that we would expect by chance and actually probably reflects the need of the immune system to be able to disseminate information quickly across all its different components. So having characterized the network, we next wanted to see whether we could do some kind of preliminary analysis to improve our prediction of how immune cells will respond to a particular stimuli. So our goal was to predict the strength of a response of different immune cell types to stimuli using immune network structure. We hypothesized that cells directly activated by a given stimulus would be the most strongly activated, with decreasing activation scores as the distance between a cell and stim increases. So to do this, we tested out 3 different stimuli that hit different parts of the immune system. We took immune cells from mice, isolated the immune cells from the blood, ran CyTOF on them to both characterize the cells themselves as well as their activation status. And we found that the first part of our hypothesis was true in that cells directly activated by stimulus are the most strongly activated. So here, the stimulus is up at the top. This is just an example of one of them, but it's representative of the pattern that we saw. The immune cells in the first row are those that are only 1 step away, and then 2, and then 3. The size of the node indicates the number of shortest paths, whereas the color indicates the activation score. So you see here that the cells that are just 1 step away, directly activated, are the most strongly activated. But we didn't see the stepwise decrease in activation that we expected. And you can see here that the length of shortest path, once you get past the cells that are directly activated, does not correlate very well with the activation score of the cells. So we decided to look at something else. So over here is a schematic showing that if you have 2 cells that are both 2 steps away from a stimulus, the shortest path is 2 steps, but if there's only 1 way for cell A to be activated in 2 steps and 3 ways for cell B to be activated in 2 steps, then we say that cell A has 1 shortest path while cell B has 3 shortest paths. And so if you look at the number of shortest paths in relation to activation score, you see that it is quite positively correlated and that this correlation is significant. And this shows us that we're better able to predict how an immune cell will respond to stimulus if we have prior knowledge of its place in immune network structure. So this is kind of a proof of concept of the value that this formalized knowledge of immune interactions can provide. So to summarize, ImmunoGlobe as a computational resource reflects known features of the immune system and enables generation of novel insights from experimental data. The graph structure also allows application of network analysis methods to immunology for the first time. As a public resource, this network interactive is available at immunoglobe.org. There's a user interface where you can explore the interactions, you can download the cytoscape file, and also download the graph and run your own analyses. So thank you. If you have any questions or would like to discuss, please feel free to email me.